Hello everyone, my name is Samuel Longoria and I will be presenting the project titled Theranostic Nanosystems with Synthetic Peptides for the Treatment of Prostate Cancer at the Genome Level. Cancer is one of the diseases with the highest mortality worldwide. In 2020, prostate cancer was uh, ranked in second place just below lung cancer. In Mexico, prostate cancer ranked first by its incidence and fourth by in mortality as well in 2020. One of the key genes that can lead to a cancerous phenotype is MYC. MYC is a critical transcriptional factor in cell development and reprogramming towards a cancerous phenotype, and it has already been seen that its level is above average in up to 70% of cancer types. The main function of the MYC transcription factor is the regulation of various essential processes such as DNA synthesis, mitochondrial and ribosomal biogenesis, as well as glutaminolysis. MYC acts through heterodimerization with a complementary protein called MAX. This heterodimer binds to regions in the DNA that are called Ebox, thus allowing the transcriptional activation of genes. During cell reprogramming towards the cancerous phenotype at elevated levels of MYC protein, MYC binds to Ebox sequences when they usually would not, increasing gene transcription and transforming the normal cells into tumoral cells. In literature, this phenomena is called promoter invasion. And according to DepMap portal, there are several cancer cells or cell lines that have an expanded or above average MYC expression. One of them is prostate cancer. However, there are several other lines compared to the baseline level of 0. Following the DepMap portal, we have the transcripts per million, and we can see in cancer cell lines there is a higher amount of transcripts per million in MYC expression compared to the prostate in healthy males, according to data from the Human Protein Atlas. This means that most cancers will have an elevated expression of MYC. Now, going into the methods of this work, first there was the design of several peptides derived from MYC and MAX proteins. For this, we use several bioinformatic tools such as Swiss model, CN3D, and blast protein for this peptide design. In addition, the Scoop tool was used to calculate the protein stability curves as a function of temperature. Some general chemicals that were used in this work, we have the gold nanoparticles conjugation kit of the cytodiagnostics brand. We have also a phosphate buffer or PBS at a pH of 7.4 that was used to dilute the peptides. And the peptides were synthesized by Acura Monterrey in the state of Nuevo León. Spectrophotometry of UV visible was used to gain the absorption spectrum using a nanodrop 1000 of the Thermo Fisher Scientific brand, and the absorption measurements were carried out from 220 nanometers up to 750 nanometers at 25 degrees in PBS at pH of 7.4. We also carry out a hemolysis test in order to determine the effect of polymers on erythrocyte lysis. The protocol following was based on the previous report by Pinilla Torres and colleagues in 2021. We also carry out a cytotoxicity MTT assay to quantitatively determine the effect of cellular proliferation, viability, and cytotoxicity of these nanosystems in the cell line LNCAP. The protocol following this work was based on the previous reports of Roacho Perez and colleagues in 2021. So we developed 4 different nanosystems, we functionalized 4 different gold nanoparticles. The first one is gold nanoparticles with the MIC peptide. The second one is the gold nanoparticles with the NLS, or the nuclear localization sequence, that was used as a reference. We also functionalized gold nanoparticles with the MAX peptide. And finally, we functionalized gold nanoparticles with a mixture of MIC and MAX peptides. Okay, so now going into the results, we have the spectra of the nanosystems. First, the design peptides have a mass of approximately 8 kilodaltons each. The MAX peptide has an isoelectric point of 10.45 and a charge of 4.24 at pH 7, while the MIC peptide has an isoelectric point of 10.61 and a charge of 4.95, equally at pH 7. The mix melting temperature or protein stability was calculated to be at 80.1 degrees, while the max protein stability was up to 81.4 degrees. Now, going back to the spectra, we can see there's a displacement in the wavelength of the maximum absorption point of each of the different nanosystems. In red, we have the gold nanosystems, the unfunctionalized or non-functionalized nanosystem, and the maximum absorption point was at 517 nanometers. Once these gold nanoparticles were functionalized with the different peptides, there was this displacement or an increase in the wavelength that the nanoparticles have the highest absorption. We can see in the case of the MIC max functionalized nanoparticles, it was at 523, 526 for the nanoparticles with the peptide MIC, 523 for the nanoparticles with the NLS peptide, and once again 526 with the nanoparticles with the MAX peptide. The functionalized gold nanoparticles have a displacement of 9 for MIC and MAX peptides compared to the non-functionalized gold nanoparticles. While for the gold nanoparticles with the NLS peptide, this displacement was of 6 nanometers. However, it is important to note that this could be especially because the NLS peptide has a much smaller mass. It is of 1.3 kilodaltons compared to the 8 kilodaltons of the MIC and MAX peptides. Regarding the hemolysis test, we tested 3 different concentrations: 5 micrograms per milliliter, 0.5 micrograms per milliliter, and 0.05 micrograms per milliliter. At 5 micrograms per milliliter, non-functionalized gold nanoparticles and functionalized gold nanoparticles with MIC and with NLS were found to be hemolytic, which can be seen in the figure with a percentage going from 2 to 5%. At 0.5 micrograms per milliliter, the non-functionalized gold nanoparticles and the gold nanoparticles functionalized with NLS were still considered hemolytic, while finally at the 0.05 micrograms per milliliter, the gold nanoparticles functionalized with NLS were still considered hemolytic. This could be especially or specifically by the non-specificity of the NLS peptide, having an effect on the cell membrane of the erythrocytes. Finally, into the MTTSA, the 3 previously mentioned concentrations were used, and at these 3, the functional gold nanoparticles with MIC and with NLS were considered cytotoxic. We can see this difference on the figure, where we have in green color the results in cell viability for the gold nanoparticles with MIC in its 3 different concentrations, and in red the gold nanoparticles functionalized with the NLS peptide. However, there was no evidence found of cytotoxicity or cytotoxic activity in the non-functionalized gold nanoparticles and the max functionalized gold nanoparticles. In conclusion, nanotechnology presents an opportunity to develop theranostic nanosystems, that is, combining diagnosis with therapy, since the construction of a nanosystem can be carried out with a molecule to diagnose the disease as a coupling, as well as coupling a therapeutic molecule. Furthermore, synthetic peptides allow selecting desired properties and excluding undesirable properties of proteins. The synthetic peptides used in this work, by binding to the DNA of tumor cells, prevent transcription, which leads to cell death and therefore having a therapeutic effect in cancer. Studying the interaction of this nanosystem with the genome allows identifying genomic signatures that will provide information on the mechanism involved in cell death, such as apoptosis, necrosis, amongst others. These preliminary results provide a scaffold for further testing using this synthetic peptides design. We will evaluate MYC-driven cytotoxicity in other prostate cancer cell lines, such as VCAP, DU145, and PC3. Cancer cells with high MYC expression should be sensitive to MYC peptide, and in a non-cancerous cell line, the MYC peptide may not present cytotoxicity. The present study was funded by the Consejo Nacional de Ciencia y Tecnología in its call for basic scientific research with this grant number. And in behalf of all the nanomedicine lab members, we would like to thank you for your time. And if you have any further questions or any inquiry, you can contact me at the following email address. Thank you.